Oscar the elephant was a very special organism in Africa. He loved life in Africa, but he always did wonder what made his skin color gray. So he knew he would visit the great emperor monkey to find out. The monkey started giving an explanation. Just to get started, transcription is the process of making an RNA copy from a DNA template. Throughout the process of transcription, the monkey stated, there are three major steps: initiation, elongation, and termination. The first step of transcription is initiation. In initiation, RNA polymerase binds to a promoter on the DNA. These promoters signal the beginning of a gene. The second step of transcription is elongation. In elongation, the RNA polymerase makes a complementary RNA strand from one of the DNA strands. This DNA strand is called the template strand. The complementary RNA strand has codons in it, which is 3 consecutive bases that specify a single amino acid to be added to the polypeptide chain. They are basically what make the polypeptide chain longer. The third and final step of transcription is termination. In termination, the RNA polymerase comes across a DNA sequence called a terminator and stops the transcription process. One of 3 stop codons, UAG, UAA, or UGA, stop the transcription process. The mRNA that was made during transcription inside Oscar the elephant first must get processed before it can leave the nucleus. First, the introns that do not code for anything must be cut out. Then, the Then, the exons that code for a protein must be spliced together. After that, the methyl cap and poly A tail are added and the mRNA is ready to go to the cytoplasm to be translated. And the mRNA code must undergo a process called translation. Translation is the process in which a ribosome reads the mRNA and makes a protein. mRNA, tRNA, and rRNA are all needed for translation to occur. mRNA uses a start codon called AUG to signal that it is ready to be formed together. Each transfer RNA has an anticodon whose bases are complementary to a codon on the mRNA strand. The ribosome positions the start codon to extract its anticodon. The ribosome also bonds the next codon and its anticodon. Then, the ribosome joins the 2 amino acids and breaks the bond between methionine and its tRNA. The tRNA floats away from the ribosome, allowing the ribosome to bind to another tRNA. The ribosome moves along the mRNA, binding new tRNA molecules and amino acids. These processes continue until the ribosome reaches one of the 3 stop codons. The result is a complete polypeptide. The emperor monkey stopped his discussion and was amazed when he glanced at Oscar the elephant. Oscar had turned green. Oscar was scared at what had just happened. The emperor monkey reassured him that he was alright. He had just gone through a gene mutation known as substitution. Substitution is when one base is replaced with another in a particular gene. Substitution only affects a single amino acid in the protein. Substitution changes the shape of the protein and gives it a new function, hence the new green color on Oscar. In Oscar's case, the gene mutation was not harmful, but sometimes it can be. Oscar then thanked the emperor monkey for his time and then trotted away in the night.